Hi, everyone, and welcome to Seed Speaks. This is a video series brought to you by SeedWorld Group. My name is Marc Zienkiewicz, and I am an editor with SeedWorld Group. And this month we are talking with experts about the field of agronomics. I'm joined today by Gil Ronen, founder and CEO of Energene, Genevieve Arsenault-Labrec, co-founder of EOS Technologies, and Kim Kenward, R&D manager for 2020 Seed Labs. Welcome, everybody. Good to see you. Today we're talking about why DNA is an agronomic game changer. Not just DNA, but its associated technologies. Now, you all have something in common, and that's the fact that your respective companies were recently on Germination magazine's list of top 10 innovations for 2022. Congratulations. Before we talk about that, though, I'd like to talk a little bit about DNA for a minute. Let's go around the table and get your take on this question, how DNA and technologies related to it are changing the field of agronomics. Genevieve, I'm going to start with you. What are the thoughts on top of your head when you hear that question? How is DNA and related technologies changing the field of agronomy and agronomics?
Yes, but on my side, if I think about disease diagnostic, what I'm working on, if we only look at PCR tests, where you can identify a pathogen just by using a DNA sample, that brought a new level of diagnostic that allow first to get rapidity. So you can get the results from a sample in one day instead of several days, for example, to isolate the pathogen before to identify it. It also brought a new level of sensitivity that allow, in part, early detection. So you can find the pathogen before even seeing the symptoms, or you can identify an organism that cannot be detected easily by isolation. One other thing that is nice about the different technologies we see today is that we are not only able to detect the presence of a pathogen, if it's there or if it's not there, but we are able to identify different characteristics from this pathogen, its species, its virulence profile. And I think that this level of precision will allow agronomists and growers to take specific actions.
Kim, what about you? What comes to your mind when you hear that question? How is DNA changing the field of agronomy?
Well, I see agronomists actually performing an incredibly important function out in the field, and they're on par with what I see a seed analyst doing, in that they can go in and they can identify a huge amount by eye. It's like they're driving down the highway at highway speeds, and they're identifying which field has which crop going in it, and then they pull into the one that actually is showing stress signs, walk in, find the point that you've got stress, and then they're examining the landscape, the roots, the leaves, and they're actually figuring out what they think the problem is and what is the most likely cause of that problem. But there's a limit to what you can do visually. You have to have symptoms of some sort, you're still looking at a very accurate, knowledgeable deduction rather than an absolute diagnosis. So molecular comes in and fills that gap. So it provides the data that sort of says, this is exactly what it is. And then it makes the jump so that you can actually do it before there's symptoms in terms of what you see happening with the plants. So it gives you that early warning that you don't have when it's just a straight agronomy decision.
Last but not least, Gil, what are your thoughts on that question? How is DNA and its associated technologies changing the field of agronomy?
First, thank you for inviting me. We are working with Energene, over 10 years on developing new varieties of all different crop plants by ourselves or together with our partners. And if you think of different varieties of crop plants, like say, let's take tomato, between a processed tomato used for ketchup and a yellow cherry tomato, the difference is in the DNA. Both of them are naturally bred, both of them bred for very different uses, and the difference is in the DNA. If you are capable and able to identify those differences, you can develop better performing varieties in a very short time manner and a cost-effective manner. If you don't have that, it will take double the time. It can take 10 years to find a better performing variety, and it's more time, more money, and we all lose from having less of the best varieties to consume as food. So this is for us, when I hear DNA, it's about the DNA data that enable us to bring better performing varieties for farmers and for everyone.
Now, back to our top 10 innovations. Genevieve, AOS Technologies is marketing a new molecular test in Canada that uses genetic markers to identify quickly and precisely which variants or pathotypes of Phytophthora root rot are present in a grower's field so that proper resistant varieties can be used in the following year to fight the disease. That's pretty significant. Can you tell us about this in a nutshell, how it works and why it's so significant?
Yes, in fact, every year in North America, growers are losing $500 million due to Phytophthora root rot. Do you hear me well?
Yes, you're coming through loud and clear.
Okay, good. So the best way to fight Phytophthora root rot is to use soybean varieties that contains one of the 5 resistant genes that are found in the market. There is 5 main genes, but to know which variety to use with the right genes, you need to know which variants of the disease you have in your field. It just in Canada here, because growers did not have this information in the past years, 85% of growers struggling with it were not using the right variety with the proper resistant gene, while this option was there under the ice. Our tests allow from AOS Technologies, our tests allow from one simple soil samples to detect exactly which variant of Phytophthora is present. So we can then recommend exactly which varieties should be used for the next season. We think this is a game changer, as using the proper variety will get growers full protection from growing to harvest without even adding any input to the fields, and just by making the right choice.
Now, Kim, 2020 Seed Labs has developed a new test that provides a needed tool for Canadian seed growers and producers to avoid soybean cyst nematode transfer between fields with planting seed or machinery and monitor the effectiveness of resistant varieties. How does this test work, and why was it needed, and why does it matter so much, most importantly?
So soybean growth in Canada is a relatively new crop for the Western Canada. And we deregulated soybean cyst nematode in 2013, even though we know that it's endemic within the United States. So we have had reports of it in Canada before, particularly in Ontario, but there's always been a debate about whether or not it's established. But now that we have seen the deregulation and we're now increasing our level of acreage that are being grown because of the early maturing varieties, we're starting to expect to see the soybean cyst nematode show up. So it wasn't a huge surprise that when we saw it turn up in Manitoba, and then we actually starting to recur. So we've got a really early infection level starting to happen within Canada, but the tests available for detecting soybean cyst nematode are really based on what's come out prior work happening in areas that you had huge loads of it. So, and if we're going to actually manage the disease within Canada, we need to have an early warning tool of where cyst nematode might be showing up, and it needs to be workable before you actually see symptoms, because we know that at early levels of infection, we can get up to like 30% loss on the soybean yield because of the soybean cyst nematode, but we haven't actually seen any symptoms of it in the field yet. So this is meant to be an early diagnostics tool that will help us monitor and quarantine fields if needed on a field by field basis, so that we can actually manage the disease before it becomes really big.
Now, Gil, Energene developed its SNPeer software as a customized genotyping solution in which genotyping and imputation are optimized within an individual breeding program to maximize the genetic data gained from a minimal number of SNPs genotyped. So, for those not familiar with this, SNP genotyping is the identification of diversity and genetic variation that exists within a given DNA sample. Gil, can you talk a little bit about how it works, why there was a need for this software, and how it could change the field of agronomy for the better?
Sure. So when we develop new varieties, we are using the genetic diversity that exists within a given crop, and this reflects hundreds of millions of differences in the DNA between any two individuals, and it's very hard and expensive to detect them all. So what we do DNA tests that is sampling the DNA, let's say 1 to every million differences across the whole genome, and then we can sample the whole DNA in a very cost-effective manner. But we do want the whole information. We do want to realize all the hundreds of millions. So this is exactly what our technology is doing. So we actually sampling, let's say, hundreds of points in the genomes, but because we know the whole information of the founder lines, all the information that exists within the different varieties, we can on the computer fill in the gaps between those hundreds and come up with hundreds of thousands of even the whole genome and the whole differences. This is very important because if you do, let's say, wheat now has a lot in headlines right now. If we are doing wheat breeding in Europe, it's very different from wheat breeding in Canada or in the US because the varieties are different. So we can, if we are using the same one type of DNA test, it is still very expensive and it's not reflecting the differences between every given material. So what Energene is doing, we do a tailor-made test for every breeding program. Even if you do wheat breeding in Canada and you have 5 different breeding programs, you can use 5 different DNA tests. This is minimizing the overall SNPs that are tested, and this is minimizing the overall cost, but the imputation gives you the full picture. So this is the idea, minimizing the DNA analysis cost, the genotyping cost on one end, without losing any of the information that is the background. So this technology is well adapted and is generic, so it fits all crops and animals. And because it's tailor-made, we don't need any historical information or any public information in order to build for every customer and every project its own DNA test.
Now, you have all helped develop some important new products that, in their own way, help advance the field of agronomy through analyzing DNA and exploring the genome. Thinking about the future, what excites you the most when you think about how your work could alter the field of agronomy for the better? Let's gaze into our crystal balls here for a minute. This time I'll start with you, Gil. We'll go in reverse order. Gil, looking at the decades ahead, what could the field of agronomy look like thanks to the insights breeding programs can gain by using your technology?
So today, when the varieties of any crop, wheat, tomato, soybean, is developed, it developed to key geographies, and one variety is used by many farmers that in different territories within the given geography, there are different cultivation conditions, different environment. So we don't believe that in the long run, it is the best use or is to have a single variety for big geography. So what we believe that our technology will enable companies, seed companies that develop new varieties to have more tailor-made, more niche varieties that fit specifically different farmers that use in different geographies or different niches, different cultivation protocols, etc., whether they're irrigating artificially or not. So for every farmer, there will be more options of varieties, and he or she can identify the best suitable for their needs according to the DNA test that we provide.
Now, it's amazing for me to think about how we could one day conquer soybean cyst nematode, Kim. That's a pretty neat thought. How might a technology like the one that 2020 Seed Labs has developed help us get there?
Well, conquer is a really big term. I don't think we're ever going to be able to eradicate it like we did polio. But I think we can actually get to the point that we might be able to live with it and manage the yield losses associated with it. So what I see is that the old test that was available for soybean cyst nematode was actually developed in a area where you actually had a lot of it already present. So it desperately needed an update because it was missing a lot of the this earlier life stage forms or the dormant life stage forms, because it was really primarily focused on the living motile forms. So our test was basically designed to try and increase the sensitivity of the capability of detection so that we could spot it at an earlier stage. And if we can spot it at an earlier stage, then we can actually see it as a potential problem before you start seeing the large yield losses. So ultimately, what I would really like to see is soybean in particular becoming a much better and stronger crop option for more areas of Western Canada, so that farmers have another alternate choice in terms of what they can grow.
And Genevieve, I'll give you the last word. Could we one day defeat Phytophthora root rot thanks to this technology that EOS Technologies has come up with?
I don't know if we will eradicate the Phytophthora root rot, but we can undeniably lower the pressure that is put by Phytophthora on soybean production here in Canada. I think we can also reduce significantly the population level that is found in the soil by using year after year the right varieties. But for us at IS Technologies, what is important for us is that we know that we will be able to preserve for a longer time sources of resistance that are carried by soybean varieties and that has been developed through important breeding efforts. Because what we observed in the past and still today is that some of these sources of resistance are used extensively and repetitively, and this is used for several years, and that led to the declining efficacy of some of these resistant genes that are not efficient anymore. So we think that today is the right momentum to use the proper variety based on the proper variant you find in your field to ensure the durability of these, the sustainability of these sources of resistance for the future. And it also promotes a method to curb the disease that is sustainable for the environment and eliminate the dependence on chemical input.
Now, before we sign off for the day, we have an audience question. There's so many pests, diseases, etc. around the globe causing issues in terms of agronomy. How do you all target what to focus on in terms of breeding? How do you make sure you're testing the right things? Let's start with Kim this time. Kim, how do you make sure that you're focusing on the right things? I mean, there's so many factors here, and how do you know that you're honing in on what you should be honing in on, so to speak?
We don't actually work on the breeding side, although we have helped out breeders in terms of working in terms of identifying when a pathogen is present, and you can actually use molecular technology to measure the relative amounts of infection that are happening within the plant to help support breeding programs. From our perspective as a seed testing lab, our interest is primarily in terms of dealing with what's already in quarantine or we suspect is needed to be surveyed to make sure that we can see the early signs of something happening. And the idea is that you're buying time for the breeders to actually come in and be able to find those resistances and make them work for you. so that you can do things like you can select the correct variety based on what resistance is actually present in your field. So we're already seeing that with something like blackleg. But with the Phytophthora project that Genevieve's talking about is another one of those things where you're actually moving beyond just straight detection to matching up the breeding information with the pathogen so that it helps an individual growers.
Genevieve, how do you narrow down your priority list, so to speak?
But I agree with the questions. I was thinking about it when you asked me how DNA changed the face of agronomy. I think that with technologies like next generation sequencing, where you gather a large amount of data, you can do metagenomic analysis where you identify all the microorganisms you have in your soil. I think the challenge after that, when you get the data for agronomists and growers, is to focalize, is to make sense of these data and not get overwhelmed by the huge amount of data that they are using. And that's what we do also when we do research. We have this possibility now of getting so much data, and I think we need just to listen to the growers and the agronomists to know what to focus on.
Gil, the last word. How do you manage to make sure you're focusing on the most important things?
Yeah, so unfortunately, we all know what to focus after it made the highest damage. So we focus on the most damaging diseases and the most devastating diseases. And this is as I said, unfortunately, the best way to realize what we need to focus on. For example, now in energy in Canada, we focus in canola on the clubber disease. This is a fungal disease that is a very most devastating disease. It causes a lot of damages now in Alberta, but moving to Saskatchewan and beyond. On the other question is how fast do we react? So the moment we realize there is such a disease spread, so we can try to avoid it, try to eliminate, try to mitigate, but the best way is to develop better performing varieties that will naturally resist this disease and then avoid the loss of yield, etc. And this is exactly what we do. So we try to cut the breeding times by half. So for canola from 4 years to 2 years, and then come up with new varieties faster that will, so the next year the damage will be way lower. So of course there are DNA technologies to sequence the soil and the plant and identify the diseases, but it's still very hard to realize from the sequence what is the actual damage to the plant. So we need the agronomists, we need the farmers to tell us what hurts most and react accordingly.
Fascinating insights, everybody. Thank you so much for your time today. That is all the time we have. I just want to thank our guests, Kim and Genevieve and Gil, for joining me. To check out their innovations on Germination's top 10 innovative products list, visit germination.ca/innovations. Join us next week when Ali Roden tackles the topic of managing abiotic stresses during pollination. Genevieve, Kim, and Gil, thanks so much for your time.
Thank you very much.